In this session today, we look at some of the more conventional forms of treatment for cancer, starting with either radiation, chemotherapy, surgery, or adjuvant therapies. Let's look at each one in turn. So surgery is still the first point of treatment for most solid tumors, and there are advantages and disadvantages for surgery. The advantages are that it's quick and effective. It still accounts for the largest number of cures that we see for cancer. And also at the end, pathological sections are done to confirm that the tumor has been excised completely. The disadvantages are that guaranteed removal is not always the case. You do get some tumor cells which escape. Also, depends on the location of the tumor. For example, if it happens to be in the brain, it's much more difficult to take off all the tumor tissue without damaging some of the surrounding critical normal tissues, which can affect function. And lastly, it's also ineffective for spreading tumors or metastasis. One of the more commonly used forms of therapy is by giving it radiation. And the source of radiation can be either external or internal. Internal radiotherapy typically uses a radioisotope, which could be, for example, in a ferromagnetic bead, which is implanted directly in the tumor. Most commonly, this is used for prostate cancers or bladder cancers, and the idea is that you get localized radiation to the tumor itself. External radiotherapy relies on external sources of radiation like X-rays or cobalt rays, and the idea is that these gamma rays are used to blast the tumor. Whatever the source, both forms of radiation have the same principle in how they destroy cells. They work by radiolysis. What that means is that radiation creates unstable radicals called free radicals, which target either DNA or the components of the cell indirectly or directly, and this results in the breakdown of the DNA or breakdown of the cells, and that kills the cells. So radiation as a generic source can be refined in order to localize radiation beams just to the tumor and avoid the surrounding normal tissues, and this is called as conformal radiotherapy. In this case, imaging techniques are used to localize the tumor first, and then the radiation beams are targeted in such a way from various sources surrounding the body, in such a way that the focus of the beam comes only to the tumor and minimizes radiation damage to the surrounding cells, normal tissues. This is called as conformal radiotherapy. Using this mode, you can increase the amount of radiation that can be delivered, and also you can get precise and focused delivery. And in some liver tumors, this has been successfully used to deliver up to 60 gray of radiation, which is a large amount only to the tumor. Other ways of using conformal radiotherapy is gamma knife or CyberKnife, which is used for brain tumors. And again, it's like a cap that is fitted onto your head. And these beams of radiation surround the head, but the focus of the beam is only to the tumor within the brain. Another way of using conformal radiotherapy is fractionating the doses. Usually radiation is never given in one single dose. Hyperfractionated radiotherapy gives multiple doses of radiation, smaller doses, but lots of them over a period of time. And the idea behind this is that if any cells escape radiation in the first round, they are targeted in the second round. So by the time they start to divide again, the second round of radiation will hit them, and so on and so forth. And the idea is that you kill as many tumor cells using fractionated doses instead of one single dose. Like with most forms of therapy, there are pros and cons to using radiotherapy. On the plus side, it's again very effective, it's quick compared to some of the other forms of treatment. On the negative side, there are lots of side effects associated with radiation, including vomiting, hair loss, and radiation-induced tissue necrosis or damage to normal tissues. There's also the chances of relapse because not all the tumor cells might be targeted by radiation. And lastly, There's also an increased risk of metastasis because you're dislodging the tumor cells and chances are they would colonize a second site. Adjuvant therapies combine typically both radiation and chemotherapy and other drugs to maximize the effectiveness of treatment. And one form of adjuvant therapy uses bioreductive drugs. Let me show you an example. Now, what happens in typical solid tumors is that you get subpopulations of cells which are hypoxic. As you can see in this figure, this is an actual tissue from a rat tumor. And as you can see, the staining in red is the blood vessels. The staining in blue are oxygenated cells, so tumor cells that are very close to the blood vessels, so they have enough oxygen. They're called oxic cells. But as the tumor grows bigger in size, a subpopulation of cells, as they move further away from the blood vessel, they become hypoxic. And these hypoxic cells shown in green are dangerous because they tend to avoid or they are resistant to radiation and chemotherapy because they're not actively dividing. They're alive but not dividing. And as a result, when the radiation and chemotherapy is finished, these hypoxic cells start to divide again and recur, and therefore you get relapse of tumors. So one clever way of targeting both hypoxic and oxic cells is using drugs called as bioreductive drugs, which is a part of research I was involved with. Bioreductive drugs target only hypoxic cells. So in normal cells, oxygenated cells, they're completely harmless. But when they encounter a hypoxic environment, these are converted into cytotoxic prodrugs that kill the cells. Bioreductive drugs are clever drugs that only kill hypoxic cells. So under normal oxygenated conditions, these cells are totally non-toxic. But when these drugs encounter a hypoxic environment, they are converted into a cytotoxic compound, and this targets and kills the hypoxic cells. So using bioreductive drugs in combination with radiation or chemotherapy is a very effective way of ensuring that both tumor types, hypoxic and oxic cells, are killed off. So chemotherapeutic drugs are essentially drugs that affect tumor cells and stop them from dividing and instead target them for cell death. And they're non-selective in their target cells. So chemotherapeutic drugs, what I've shown you in this slide is simply an overview of how these drugs work, starting off with the red alkylators at the top. Alkylators, as the name suggests, add alkyl groups to DNA. And what happens as a result is that DNA forms knots. And it cannot replicate, it cannot make copies of itself, and it cannot make proteins. Alkylators work by forming adducts in DNA. The next category are inhibitors. Inhibitors inactivate proteins or DNA inside the cell. So typically there might be key enzymes involved in replication, but they inhibit the activity of these enzymes, thereby affecting DNA or protein function. The next category is toxins. Typically, these are natural compounds derived from natural sources, and the example I've given you here is taxols, which is derived from the yew tree. Now, taxols typically affect a protein called tubulin. Tubulin is a key cytoskeletal protein. What it does is, tubulin is needed for giving structure and shape to the cell, and it's critical at the time of mitosis or cell division. So toxins like taxols prevent tubulin from working and the cell cannot divide properly. The next category is antibiotics, which you may be familiar with in terms of bacterial infections. But antibiotics, some of them can also be used in tumor treatment. And the way antibiotics work is that they inactivate proteins or DNA inside the cell. They prevent the activity of certain enzymes, for example, topoisomerases, that are needed for replication of DNA. And the last category is antimetabolites. Antimetabolites are drugs that target key metabolic pathways for DNA synthesis. For example, enzymes that are needed to make or synthesize new DNA. These drugs target those antimetabolites. So the Main take-home message from this session is to understand that conventional forms of therapy like radiation and chemotherapy and adjuvant therapies target cells, and typically it's like you can use ordinary house bleach even to kill tumor cells. So the problem is that these conventional forms of therapy are not selective. They don't only target tumor cells, but they also can kill normal cells in the process.